Hello everyone, and welcome to today's webinar in the Public Health Webinar Series on Blood Disorders, presented to you by the Division of Blood Disorders at the Centers for Disease Control and Prevention, also known as the CDC. My name is Donna DeMichele, and I'm the Deputy Director of the Division of Blood Diseases and Resources at the National Heart, Lung, and Blood Institute at the NIH. And I'm pleased to be with you today to serve as the moderator for today's topic entitled My Life, Our Future, Advancing Hemophilia Care Through Genomics. But before I introduce our guest speaker, Dr. Barbara Konkle, I would like to go over a few points and helpful tips for joining this webinar, for those of you who may still be in the process. So, upon joining the webinar, you'll see two audio options, and you can opt to listen via the speakers on your laptop or by using your phone to call in. If your phone option is chosen, the webinar tool will provide you with the phone and access numbers and the audio pin, which you'll need. Please also note that all audio will be muted, so that at the end of the presentation, you will have an option for questions, not during the presentation, but at the end of the presentation. And to submit, all questions will be submitted as written questions. And to submit a written question, please use the questions area of your toolbar. And we were going to try to get to as many of your questions as possible, and the presenter will respond to as many of them as the time allows. Today's webinar will last approximately 1 hour. It's being recorded, and it will also be available to you subsequently, and I'll tell you a little bit more about that. But before we begin today's webinar, we need to start with an audience poll, since the organizers are very interested in knowing a little bit more about you. So let's proceed with the poll. So for question number 1, where do you reside? So most of you are calling and listening in from the United States, but we also welcome our participants from outside the U.S. So tell us a little bit about and what you do. So looks like most of you provide direct patient care to persons with bleeding disorders, but that there are many of you who work in other settings, and we welcome all of you. Thank you. So among those of you who provide care to bleeding disorder patients, in what setting do you do so? So many of you, or most of you, are on staff at hemophilia treatment centers. But some of you are not, and we welcome one and all. So thank you all for listening, and thank you also for those of you who are persons with hemophilia or bleeding disorders who are listening in as well. So now it's my pleasure to introduce today's presenter, Dr. Barbara Konkle, who wears many hats. Dr. Konkle is the Associate Chief Scientific Officer and the Director of the Hemostasis, Platelet Immunology and Genomics Laboratory, the Associate Director of the Washington Center for Bleeding Disorders at Bloodworks Northwest, and a Professor of Medicine at the University of Washington, all in Seattle, Washington. She's had over 30 years of experience in research, clinical laboratory testing, and clinical care for patients with inherited bleeding disorders, including, of course, hemophilia. She has active U.S. government and industry-funded studies and has published widely in her areas of expertise. Most importantly, for the purposes of this presentation, she is the principal investigator for the National My Life, Our Future project, which she will tell you about. This project has now genotyped over 11,000 persons with hemophilia and also potential and confirmed genetic carriers. So I am going to now welcome Dr. Konkle to begin her very exciting presentation. Welcome, Barbara.
Thank you. Thank you very much, Donna, and for the organizers for giving me the opportunity for this presentation. So what I'd like to do in about the next 40 minutes is first provide an overview on the role of genotyping and its limitations in answering research questions in hemophilia, and then bring you up to date, describe the structure of the My Life, Our Future program and bring you up to date on where that stands, share some results, and also the implications for future research in hemophilia. So just a little background on hemophilia. So hemophilia A and B are due to deficiencies of either coagulation factor 8 for hemophilia A or factor 9 for hemophilia B. Hemophilia A is more common than hemophilia B, but the clinical presentation is essentially the same. These are X-linked disorders affecting somewhere between 1 in 5,000 to 1 in 7,500 male births. The mutation rate is high, the new mutation rate is high, and estimated at about 30% for hemophilia A and B. And because of that, hemophilia is seen worldwide and in all racial groups. And this has been shown by many investigators to be due to diverse factor 8 and factor 9 DNA variants, which is the terminology we're supposed to use now, but may be known to you as mutations. So why are factor 8 and factor 9 so important? Well, this is coagulation, and factor 8 and factor 9, factor 8 serves as a cofactor for factor 9A's activation of factor 10, which then results in thrombin formation and fibrin clot formation. And deficiencies of either factor 8 or factor 9 predispose to spontaneous and trauma-induced hemorrhage. So the clinical symptoms we classify hemophilia by mild, moderate, and severe, with severe disease being less than 1% activity. And if a child is not diagnosed at birth, they present on average at about 1.5 years. And then as the hemophilia becomes more mild, the presentation is later. For those who have severe and moderate disease, if they are not treated prophylactically with factor infusion or other therapy, they have spontaneous joint and life-threatening bleeds. And all patients are at risk of trauma and surgery-induced bleeding. It's a very exciting time in hemophilia in terms of treatment. We have advances in coagulation factor replacement and much progress in gene therapy and new alternative therapies that do not require factor 8 or factor 9 infusion. So, but we're going to switch to genotyping. And why do we want to know genotypes and how does it help us at this point and may help us in the future? So the major complication of hemophilia A is the development of a neutralizing antibody to treatment. And this occurs in about 25 to 30% of patients with severe hemophilia A and really early in exposure to the treatment product. It's most common in patients who have null variants, thus DNA changes that make it so they can't make any protein. And it's more common in non-Caucasian racial groups. We don't understand that. There's certainly genetics involved, likely, and their risk is higher than 30%. The risk is lower in patients with mild and moderate hemophilia A, but it's still at about 6.7% at 50 exposure days. The risk is lower in hemophilia B, but still present, and it's seen primarily in those with severe disease and particularly large gene deletions. And our understanding of inhibitor formation certainly requires our understanding of the contribution of the immune system. And that is, we still have a lot of work to do in that area. So another area where genotyping can help us is in understanding variability in bleeding symptoms. So generally they correlate with factor activity, but there is variability within each severity type. And it's important that we're able to predict the severity, because that would impact the treatment regimen, because we want to prevent bleeding, particularly joint bleeding, which once established will continue over the life of the patient. And so being able to predict clinical severity before a patient has bleeding would be wonderful. We know that there's probably an impact, and it makes sense, of the co-inheritance of prothrombotic variants, and that this may result in a milder phenotype. Also, von Willebrand factor, which stabilizes factor VIII. We know that von Willebrand factor levels affect response to factor VIII infusions, particularly in terms of half-life or how long the factor VIII stays around. And it's likely that DNA variation in the von Willebrand factor gene is important, plays a role there. And then there are, I'm sure, additional genetic variants outside the factor VIII and factor IX genes, and those that I've mentioned, that also likely impact phenotype. Another value, and this is something that early on was used to diagnose genetic carriers. So in some genetic diseases, the carrier state implies that the female does not have symptoms of the disease, but this is not true in hemophilia. That women with hemophilia and girls with hemophilia who carry hemophilia can have bleeding symptoms. And shown in the graph on the right with the wonderful data from Carol Casper years ago shows factor VIII activity levels in females who were not genetic carriers of hemophilia, which is shown in yellow, and females with who are genetic carriers, shown in red. And while the mean or the height of the curve is 50 to 60% around normal, there are women who have lower levels. In addition, in a study by MySpac, they showed that the DNA variant was a stronger predictor of bleeding symptoms in the female than the level. Since these are X-linked disorders, we think of X inactivation. So in each cell, one X is inactive, and we think of that being random. But we know that in some families that appears not to be true, and we certainly need more studies to understand that better. And then this genotyping results in the carrier also are important in understanding and optimizing management of pregnancy and the neonate. And so genetic carrier detection is important in assessing the bleeding risk in the mother and the potential bleeding risk in the neonate, and very useful and helpful in delivery planning. There's currently controversy as to the best mode of delivery for delivering the fetus. There data from the CDC Universal Data Collection System suggested that the risk of intracranial bleeding was significantly higher in affected infants delivered vaginally, although there are caveats to that. So this remains an area of controversy, and we need to know as much as we can to help the family make an informed decision, and that would involve knowing the genetic status of the mother and of the fetus. Also, there's exciting developments, particularly from the Royal Free Hospital in the UK, in using non-invasive fetal diagnosis, so being able to draw blood from the mother and determining whether the fetus is affected. And certainly we would not be able to do that if we don't know the genotype in question. And then for the pediatrician, being able to be ready for the best neonatal management, it helps to know the whether the infant will be affected with hemophilia or not. So in terms of hemophilia genotyping, kind of the milestones that helped us in hemophilia. So in 1977, Sanger sequences, kind of the standard sequencing, was first described, and that is really determining one base pair at a time in a DNA sequence. And then in 82 and 84, the factor 9 and 8 genes were cloned, so letting us begin genetic diagnosis in hemophilia. When that began, it was interesting that about almost half of the patients with severe hemophilia A genetic cause could not be determined. And that was described, that's when the intron 22 inversion was described, and later the intron 1 inversion, where all of the DNA material is there, but it's just rearranged. So when one was looking at targets, specific targets in the factor VIII gene, they did not detect the inversion. And then there have been methods described to easily detect those inversions, and they actually continue to be described as in laboratory approaches. So the intron 22 inversion, just to show this, this is the factor VIII gene. It's a big gene that has 26 exons or coding region, and then the light area are the introns. And in the 22nd intron, there is a gene that has homology to two genes shown as H2 and H3 upstream of the factor VIII gene. And what happens, and we think this happens predominantly during meioses, where the X and Y are trying to align, and the factor VIII gene is near the tip of the X chromosome, and it may kind of flip back on itself and then recombine into a gene that can't produce factor VIII. And a similar thing happens with the intron 1 inversion. So our standard approach to genotyping in hemophilia has been in those patients with severe disease or very low percent moderate, because they may actually be severe and we're measuring a little bit of infused factor. We start by looking for the inversions, testing for the inversions first, and then if they are not found, then looking at the coding regions and the intron exon boundaries and the untranslated regions, and then confirming the genotype, determining the genotype and then confirming that. The factor IX gene is smaller. It only has 8 exons, and the standard approach is to just do the standard sequencing, determine the genotype, and then confirm it. So now we'll get to MLOF Future and why we developed the project and how we have done it. It started really with 2 surveys, one to consumers by the National Hemophilia Foundation and one to providers by the American Thrombosis Hemostasis Network, and found that both reported that only about 20% of patients with hemophilia had genotyping. And the most common reasons were cost and lack of coverage by insurers and access. And so this is the partners in the MLOF Future project, but I have to give special mention to Glenn Pierce, who was then at Biogen, which began as the financial supporter, and Val Bias, the CEO of NHF. There was really their foresight to get this project going. And then AFN, which communicates with the HTCs, collects the clinical or phenotypic data, and manages the research review committee, which I'll get to. And then Bloodworks, where I am, we do the genetic testing and analysis and also house the research repository. So why MLOF? And I told you some things, but genotyping is standard of care in many European countries, and I hope I've convinced you that it is important in our care of patients with hemophilia. And then also this has been advocated, MASAC in 1998, resolution to advocate the value of genotyping with precision medicine initiatives where we really have to understand patients at the individual level so we can make the best treatment for them. And then the Genetic Information Non-Discrimination Act made it so that there's less likely to use this information in discrimination, such as in insurance. And then in the surveys, over 75% of community members expressed a strong interest in participating, and almost all of the HCCs said they would like to offer it to their patients. So our vision was to offer free genotyping to people with hemophilia and their families, with the hopes this would improve hemophilia care by increasing understanding of the disorder and build a foundation for scientific breakthroughs of tomorrow. So the process has been that at the Hemophilia Treatment Center, the patient, or for children, their parent authorizes testing, and then the blood is drawn there and sent to Bloodworks. And then we do process the sample, perform the genotyping. And then patients are given the option, and parents, of whether to participate in the research repository. This is a written informed consent because it includes they're allowing us to use the information for research, obviously non-identifying, including whole genome sequencing and some exciting new scientific approaches. So the non-identifiable genetic data actually gets also pushed into the AFN data set, and their blood samples are stored for future research, so then they can be accessed by researchers. We also have taken a slightly different than standard approach, but this is an approach that more and more laboratories have been using for other disorders. So we use what's called NextGeneration Sequencing, which that basically you can sequence a number of genes, pieces of genes at one time, and then put them back together to analyze them. And so there are different captures that are used. They're called to capture the regions of the specific genes, and we use a capture called molecular inversion probes, and really to capture those parts of factor VIII and factor IX that we had done in our standard approach because it was pretty effective in diagnosis. But we also used the inverse shifting PCR approach to detect inversions by designing probes that would also capture the inversion. For that, the DNA is cut by a restriction enzyme and then put back together by ligation. And we designed probes that would detect that ligated area and we would be able to get sequence if the inversion was there. And then basically they're all put together and mixed together and they're sequenced and then realigned. So we, in one tube, have their factor VIII, factor IX and sequence and whether or not they have the inversion. Using this approach, one can sequence more than just 2 genes, but that's what we use to focus on factor VIII and factor IX. And we have reported, as referenced here in Blood Advances, the first 3,000 patients and also more specifics on our methods. But this shows you the variants found, and it really has not changed since we progressed, so I thought I would just use this illustration. In patients with severe disease, almost half as expected have inversions. In patients with severe hemophilia B, there are more like null variants, like large deletions or nonsense where it stops the translation. And then in mild and moderate disease, more missense variants or changes in an amino acid. This is not dissimilar. There's been a lot of genotyping done in hemophilia, and this is not dissimilar from what has been found by others. One of the things, though, that was very surprising to us, given that there's been so much sequencing, is that we continue to find variants that have not been previously reported. This was in the first 3,000, and it has continued. So it suggests there is a lot of variation in the factor VIII and factor IX genes that cause hemophilia. One part of MLOF Our Future is to return the clinical report back to the Hemophilia Treatment Center, who then can share it with the patient and family. And returning clinical genetic results has become a little more complex because we want to be sure we get it right. And so many variants found in factor VIII, there is a region of factor VIII, the B domain, that is cleaved with factor VIII activation. And there's a variation in that B domain that has been well described, not associated with hemophilia. But many variants outside of that domain are causative, but not all of them. And whenever we start sequencing a lot of individuals, we're going to find variants that are not associated with the disease. And genetic misdiagnosis has been, is more of a problem in other disorders than hemophilia, but we still have to be alert to that possibility. And so in order to help us be very careful in our return of genetic results, the American College of Medical Genetics and Genomics has come up with a classification or a way to support saying something is pathogenic, thus causing the disease, or not pathogenic. And through that, because we don't necessarily know from one DNA change whether it's a mutation, which implies it causes the disease, or a polymorphism, which implies it didn't, that we do better with grading them so that we grade them into pathogenic, likely pathogenic, or a variant of uncertain significance if we don't have enough evidence. And likely pathogenic is that we have 90% certainty that it's pathogenic. This has been a challenge, and I'm not going to go through this table, but their framework, just to share with you, uses different things, like is it present in the population, in a population database. There are tools to predict whether it would be disruptive in a protein, and that's been done a lot in hemophilia. Is there functional data? If we put this in a in vitro system in the laboratory, does it make the factor VIII not work well, or factor IX? Is it present in family members who have the disease? And that's called segregation. Did it arise newly in the family? So those are the kinds of things we take into consideration when we're considering whether something is pathogenic, likely pathogenic, or a variant of uncertain significance. Now, since we are sequencing factor VIII and factor IX on all patients, we've actually had the opportunity to find some variants that have been reported in hemophilia that are likely benign. And this is an example. These are hemophilia B patients who had a factor IX variant found, which are listed here. But we also found a variant in the factor VIII gene that was reported in hemophilia previously in the large databases. Now, this variant is actually present there's a lot of copies, or what we consider enough, in a population database called the EXACT database. So it makes us really question whether this is really causative in hemophilia. And what we've done is gone back to the hemophilia treatment centers and say, could you get a factor VIII level on this patient? And that way the factor VIII level is normal, then we can say for sure that this is not a variant that causes hemophilia, and we can kind of clean up our databases. Now here's one though, in a person with a severe hemophilia B with a variant. It has been reported in hemophilia, and it's not been reported really in the population database. So it's possible that this individual has an abnormality in both the factor IX and factor VIII gene. And these are the kinds of things that the My For Our Future project has allowed us to delve into. But as I said, it really is challenging. And one of the challenges in hemophilia is, in the past it was said that the factor VIII and factor IX genes are very conserved, and so if we find a variant, that is the one that causes hemophilia. And we now know that's not true, but because of that, there are a lot of, if you look at the reports of variants, and in practice, we've sequenced one patient in the family and not confirmed it in other affected individuals, which we need to do to substantiate our diagnosis. And just as an example, to meet ACMG criteria for likely pathogenic, that's greater than 90% chance of being causative, a missense variant or a change in amino acid in a patient with hemophilia, it needs to be absent or near absent in a population database. This computational evidence, when we predict what it would do to the protein, it needs to show that it would be harmful. And then we still, though, need either expression studies or segregation studies. So studies that show that it is present in multiple affected family members. So we are obtaining a lot of segregation data through this project, and also providing opportunity for expression studies. And I think that will help us be more accurate in our genetic diagnosis of hemophilia, particularly in those variants that some variants we call null variants, and like the intron 22 inversion, we know it causes disease, there's not a question. But in the ones where there's just an amino acid change, we have to do more work on some of those. So back to MLOF, and where does it stand? So initially, we enrolled patients with hemophilia who had factor VIII or IX activities less than 50% by the local laboratory. And then we expanded to carriers and potential carriers in 2015. Enrollment stopped in December of 2017, and as Donna stated, we had over 11,000 participants, including over 7,000 males, and so we think there are about 20,000 males with hemophilia in the U.S., so just over a third of the hemophilia population. Overall, 81% consented to have data and samples in the research repository, and the genetic analysis and reports will go back to the hemophilia treatment centers to be completed by July of this year. We also, the variant results directly populate the HTC record, the clinical manager record, and we also report the CDC has a factor VIII and factor IX variant database, as does EHAD, the European Association of Hemophilia and Allied, I'm getting it wrong, EHAD database, and they, so that we can share our findings with the community. And so this just graphs the progress since 2002. See, we increasing number of patients, and you'll notice there's this significant increase really that correlated with enrollment of carriers and potential carriers, and probably ending enrollment. But I think this suggests that there's still certainly room to continue this, particularly for our carrier and potential carrier population. We did, I think, a good job of finding likely deleterious DNA variants. They were detected in all to date, and we're still finishing the analysis, but in all but 112 male hemophilia patients, so about 1.6%. And factor VIII, 13 of those have severe, 16 moderate, and 58 mild. And we know actually a paper from the CDC documented that some patients diagnosed with mild hemophilia A actually have von Willebrand disease, so we recommend testing for that in our reports. And then for factor IX, some moderate and patients with mild disease. We hope that finding these other variants, which could be further upstream, downstream, deep within an intron, can be part of research proposals in the future, which I'll get to. As I said, we continue to find previously unreported DNA variants. To date, we have found 687 in 1,111 individuals. So the research repository is a collection of samples and data for scientific study, and it's become the largest resource of its kind worldwide. and we really are looking toward its use to advance our care of hemophilia. So what does it contain? It contains the factor VIII and factor IX sequence from our sequencing. And as I said, we have, since we are sequencing both 8 and 9 and detecting the inversion, we have all the 8 and 9 sequence. We have additional DNA, buffy coat, we can extract more DNA, serum, plasma, and RNA. And the AFN database has demographic and clinical data, and the informed consent allowed for whole genome sequencing, dbGaP deposition in dbGaP and other large databases. Also okay to recontact the patient so that if we run out of a sample, the patient can decide whether to give more, but we have the opportunity to replenish the repository. And we have an NIH certificate of confidentiality, which protects the patient from having their DNA subpoenaed for legal issues. So this is just an example of the AFN data set, some of the things we have in the AFN data set. I think many of you on the line are very familiar with this and entering that, but we have demographics, but also treatment information, inhibitor information, and other information, which it makes we can do extensive genetic analysis, but if we don't have the clinical data to pair with it to really understand the genetic information, it's not very useful. So in terms of using the research repository, it's open both to scientists at academic institutions and drug discovery companies with hypothesis-driven applications. All applications undergo a rigorous evaluation by Research Review Committee, which is managed by AFN, and the committee includes those with expertise to analyze proposals who would use these data, so genomic epidemiologists, but scientists, but also HCC providers, some of whom have that expertise, a genetic counselor, and a consumer. And the criteria for analysis, 50% is for scientific merit of the proposal, 30% for benefit to the community, and 20% for the quality of the applicant. So we had our first application cycle, and there are 7 projects, and of those 4 are on inhibitor development, which is probably not surprising and appropriate since that's a major complication. 2 to better understand bleeding, 1 in carriers and 1 in hemophilia A and B without inhibitors, and 1 looking at factor VIII clearance. And we plan a next application cycle for 2019. So we were very fortunate to have our cohort accepted in the Transomics for Precision Medicine, or TopMed program. So that's a program of the National Heart, Lung, and Blood Institute of the NIH. It's broader precision medicine activities. And the goal of TopMed is to couple whole genome sequencing and other omic data with a clinical, well-standardized clinical data. And as of January 18th, they had sequenced greater than 100,000 genomes. This is an incredible resource. And so the Modify Future cohort was accepted, and to date, we have had over 5,000 whole genomes from our project sequenced. So this will allow these data will be deposited into the NIH database and allow access for scientists, and we'll be able to link them back to the research repository. But also within TopMed, we are only the only bleeding disorder cohort, but there are several cohorts that have thrombosis as their readout. And so being able to look at variants that affect bleeding or clotting may tell us a lot of things applicable to general population. And there are a lot of examples of things like that that we can study among the cohorts that will help, should help hemophilia. So what about the future of this resource? Well, in July 2018, we are going forward with 3-member organization. Biogen, then Bioverativ, incredible support for this project, and we thank them for making that possible. But they will not be the sole supporter of the project going forward from July 2018. We will be maintaining the MLOF Research Repository and application process, and we hope to collaborate with focused research projects and explore additional partnerships to expand the My Life, Our Future research repository. So I would like to acknowledge first the HCC providers and staff and the patients and families affected by hemophilia who so generously gave of their time and samples. And then it certainly was a team effort, many of whom I have listed here, from NHS, Bioverativ, AFN. Our next-generation sequencing approach was done in collaboration with Jay Shanduri and his lab at the University of Washington. Kine has provided project management and other help, and my team here at Bloodworks. So thank you very much, and I will turn this back to Donna.
Thank you very, very much, Dr. Konkle. That was wonderful. And we will now move on to the question and answer session for the webinar. And again, all questions are going to be submitted in written format. And so if you have questions for Dr. Konkle, just go to the questions area on the webinar toolbar and write in your question. I will see it and I will ask your question. I'll ask Dr. Konkle your question. So again, thank you very much, Barbara. By the way, while we're waiting for your questions, you'll see on the screen an announcement for a National Heart, Lung, Blood Institute-sponsored State of the Science workshop. This is going to be a workshop in which the hemophilia community will come together to examine the problem specifically of factor VIII inhibitors. Dr. Konkle spoke a lot about factor VIII inhibitors. And what the community will do, and this is all aspects of the hemophilia community, scientists and persons with hemophilia and everybody in between, will be coming together to generate a national blueprint for future research that can capitalize on the MLOF program and all the other research programs that are going on so that we can actually move towards solving this problem. And that meeting is going to be here at the NIH on May 15th and 16th, and you've got the registration information there. So, Barb, there are several questions, and I'm going to begin. You had indicated that among the 11,000 individuals who participated so far, that they represent about a third of the hemophilia population in the United States. And can you speak a little bit more to that in terms of the demographic, geographic representation of this population? And can you say a little bit more about the percentage of carriers that are also represented in the database?
So that's a very good question. In the project, we had participation from 111 hemophilia treatment centers across the country. So we think we really had good representation of geographically of the patients when we looked at the demographic data in terms of racial distribution, it was very similar to what has been reported in the hemophilia data set and that. So we think we also have representation racial diversity, and that's important, as I pointed out, because that is an important question in inhibitors. In terms of carriers, I think we don't know because we don't, and we might be able to get at that number better than we have. We haven't spent time on that, but it's going to depend for each person with hemophilia, the number of genetic carriers at risk, or each male with hemophilia. When we started, the affected patients, it did not matter whether they were male or female, just if they had a low factor level. then when we expanded it, it could be any females who either were known to be affected or potentially could be affected. So I think there's still a large number of individuals who could benefit from genotyping that we were not able to include.
Okay. Thank you. The next question actually relates to something you said very early on in the presentation, and is a nod to something that the hemophilia community is well aware of, and that's the cost of hemophilia care. And you noted that part of the reason for MLOF was that genotyping in this country was very difficult to get, largely due to absent reimbursement through insurance companies, etc. So the question from our audience is, have you had a chance to look at how much money has been saved with respect to the payers by the MLOF program having provided the genotyping that it has already?
We have not done that, but that's a very good suggestion. And I think a further suggestion would be to kind of model if the genotyping helps us in predicting severity and other things which would affect treatment, how much knowing the genotype would save the insurer. That might be a good thing to do. The team here at Bloodworks, as you can see that sharp rise in participants, we had worked incredibly hard at the end of the year, and they're catching our breath, but those are great suggestions that we should follow up on.
Oh, that's great. Okay, well, thank you, audience, for that very important question. Okay, there are a few questions that I'm going to ask as a multiple-part question. I'm going to turn that into a multiple-part question. There's a tremendous amount of interest in the carriers that are being tested, and one of the questions, and this most likely relates to research, to the ability to study carriers through the MLOF research program. The question is when are the carrier samples going to be available to the broader community for research? And the other sort of related question is, as you're doing carrier genotyping, how is mosaicism, hemophilia carrier mosaicism, affecting your ability to genotype and the results of the testing?
Okay, so the carrier, so a large proportion of the samples we are currently analyzing are in females, not all of them. So that though will be finished well before the next research repository application. So that those samples will all be available at the time of the next research repository application. We're also
in 2019, right? In 2019,
right,
yes.
Right. Mm-hmm. And we're also hoping that we have another bunch of patients in TopMed, so we can get whole genome sequencing on, because we don't have a lot on the females, and that could be really helpful. The second part was mosaicism. So just to put everyone, what mosaic, so if a mutation or a DNA variant in factor 8 or factor 9, when it first occurs, sometimes it's not in all the cells. And so we might not detect it in the DNA from white cells, well, it might be in the DNA in an egg. And so so that is a caveat to counseling individuals where this is a new variant in the family. Once we've seen it go through multiple family members or go once through a family member, then that's not an issue. Now, we have detected mosaicism in patients. And what we have done to try to work this out, some of the HCCs know this, we have gone back to them and said, can you find us, we would like a sample on a sister or another person in the family to try to sort this out. And one of the examples is the mother. We have a mother of 2 sons who were part of the project who had the same genotype, and the mother was negative. So we know she is mosaic. So also, we're able to, in our next generation sequencing approach, detect, and in our standard approach to, if we see both a normal gene, the normal sequence and the variant sequence. So we are detecting that in the project and reporting that, but we likely, I'm sure we're not detecting all, and so that is a caveat to genetic counseling. What I didn't mention in kind of the standard approach, which we're also using for this, is if we don't find a variant in males, or if we find a variant in females, but they have a very low level, we also use a technique that separately looks for MLPA, that separately looks for large gene deletions or duplications. And there's some individuals where we only find it through that. And so we're taking an extra step in females. Most of them, we don't find anything. So it's likely lionization or skewed X inactivation. But we're looking to be sure there's not a second variant, which we sometimes find. So that's why we have to be try to be very complete about it.
That's very important. There's a lot of interest from the audience in understanding, and I know we've talked about this relative to the carrier samples, but about when all the remaining genotype data will be disseminated to the hemophilia treatment centers. And a corollary to that question is, the 2 important questions that you talked about how nuanced sometimes some of this data is to use to counsel patients with. What's the role of genetic counselors in hemophilia treatment centers of transmitting this information? And once it's transmitted and hemophilia treatment centers have this genotype information, how have you heard they are using it to influence clinical care? So that was a 3-part question.
3-part question, yes. So all of the results will be back to the hemophilia treatment centers by the end of June of this year. We did, as I mentioned, have an onslaught of samples. We were receiving about 150 a week, which is incredible for a rare disorder. And so we are working through those. Our team is working through those. And so that will be definitely, though, by the end of June. The genetic, in males who are affected with hemophilia, we find a diagnosis and it confirms their hemophilia. But in terms of decision making in that, it has probably a bigger impact on females and deciding how to use that for reproductive choices and also understanding their bleeding symptoms. And so I think a genetic counselor plays an important role. I alluded to the question of variants occurring de novo in a family and what that might mean in terms of, we have that caveat that in that setting, if the female tests negative, there's a little bit of may not be, she could possibly still be a carrier. And so all of those things are important to help the family understand and use the information. And that's where genetic counseling is important. We have a genetic counselor as part of the My Our Future project, Sarah Ryan, who's fantastic. And she's been working with the hemophilia treatment centers, working with them to understand who's at risk if they don't have a genetic counselor, and also interpretation of data for the hemophilia treatment center. And she has conducted a survey of how it's being used and is assembling that. So I don't know the results yet, but we will be able to share that later.
Okay, that's great. That's great. Thank you very much for that information. So the next question actually is about mild hemophilia. And one of the audience has some concern that the persons with mild hemophilia are less likely to come to hemophilia treatment centers. And so you have certainly some mild patients in the database, but how is this project potentially understudying the mild hemophilia patients, particularly with respect to, the variant analysis?
So I don't know how many milds. I mean, we have quite a few milds in the project, and they may represent patients with more severe mild disease. That makes sense, but so they have more symptoms, and so that's something else to look at in terms of that. One of the important things in patients with mild hemophilia is that there are DNA variants or mutations in factor VIII in patients with mild disease that are associated with inhibitor formation. And in those individuals, if we find one that's strongly associated, clinically, we should try to work harder to avoid exposing them to factor. So using ddAVP more, using antifibrinolytics. So that's a real example where having genotyping in a patient with mild hemophilia A would impact their treatment.
Yes, indeed. And a related question is regarding particularly novel variants. You mentioned that you are sharing the data, particularly with respect to novel variants, that you are uncovering through the MLOF program with several other registries within the United States, the CDC, and also the European registries. A question from the audience is there any plan to publish this data, again, making it available to the research community? Is that venue?
Yes, because we will publish it. We will, and we, in terms of sharing the variants, we also on our website are going to have the segregation data, the data for each variant as to what supports it, because that will change over time. So if you have 3 families who have the same variant, and in one family we can show that that variant segregates with the disease in that family, then that impacts the interpretation of that variant for all the families. And so we want to be able to share that because I think that will really strengthen our diagnoses. But these are not private data. You know, we want to be sure that particularly our precious repository samples and that we want to be sure there's good science, but this is not a private thing. And so if there are, we want the scientific community to use it. I guess that's a way to put it. If someone figures out how they want to do that, we want to hear about it.
Okay, all righty. We've just got barely a couple of minutes left. And by the way, I just wanted to say that you mentioned the MLOF genetic counselor, and there is a request for contact information for this individual. I don't know if that's possible to share with the community. But as the last question, one of the things that you talked about is the importance of genetic information for predicting, particularly with respect to inhibitors, for predicting inhibitor development or predicting the risk of bleeding. Why is this important? What are the limitations of just using the factor VIII or factor IX mutation, and how is whole genome sequencing or full genomic data going to help in predictive models of what we call phenotype, either bleeding phenotype or inhibitor development phenotype?
So the first, in the genetic carriers of hemophilia, it's critical to both have the genetic diagnosis and the level, because genetic care, depending on how the X chromosome is inactivated in different cells, You can have very normal. I think the highest I've seen is 147 in a patient who was a factor VIII level in a patient who was a genetic carrier. So one cannot assume by how normal the factor VIII level is, whether they are or not. And assessing bleeding risk, while there's intriguing data that the genotype is informative, we still go by the level. So we want to know, does the female have a low factor level and what is their genotype? If they have a low factor level, it's likely they are a genetic carrier, but if they have a normal level, they can still be a genetic carrier. So the second question in flipping, like how does whole genome sequence helping us in phenotype? And I always tell my patients, you know, it's kind of like a teeter-totter in terms of bleeding and thrombotic risk and whether you'll bleed or clot. And there are a lot of factors that impact that. And understanding, there are probably factors that impact, like in hemophilia, who has worse joint disease, who's really at risk for that, who bleeds more, what's their balance of prothrombotic versus bleeding symptoms. I alluded to what really regulates other things that impact exon activation, then determine whether the hemophilia X is expressed more or less. Lots of stuff about other genes that affect the immune system and thus affect inhibitor formation. So I think there's just lots of areas that we will never completely understand by just studying the factor VIII and IX genes, because they only tell a small part of the story of how hemophilia is impacted. And one of the exciting things about such a large study is we can take patients with intron 22 inversions with the same genetic defect, and then we can say, well, why do some of those patients develop, you know, inhibitors, some not, and we can control for that where we can't in a smaller study.
That's fantastic. So lots more information, lots more to learn, and lots more information coming down the pike. Well, I'm afraid we're at the end of our time, and I would like to thank Dr. Konkle for a great presentation. I would also like to thank the audience for your great questions. I apologize to those of you whose questions were not able to be aired, but on the next slide, I actually will show you that I'll show you. I think, Barb, you have to go to the next slide.
The slide isn't moving for me.
Okay. Well, there you go. Okay. So there have been questions about where this presentation will be archived. You have the information there on this slide. And if you have any other questions, you have contact information there for Cynthia Sayers. I just want to say that there was an additional question regarding disclosures and conflicts of interest. And so I'm sure that Dr. Konkle will send those disclosures so that they can actually be archived with the rest of her presentation. With that, I would like to finish by thanking the Hemophilia Federation of America for hosting today's webinar. And I would also like to encourage you to come back to this series. And I think I need the next slide again. When on March 15th, Michael Cassidy, who's Assistant Professor of Surgery at Boston University, will actually look at the other side, the other aspect of hemostasis and thrombosis, and we'll discuss perioperative venous thromboembolism, risk assessment, and risk stratified prophylaxis for surgical patients. So I hope you're going to look forward to that one. So once again, thank you, Dr. Konkle. Thank you, audience. Thank you, CDC, and thank you, HFA. Have a good rest of the day, everyone.
Thank you.